Okay, guys, shall we start? So last time we finished Module 3, which was about cell-water relations. Are there any questions about water potential? Are there any people here who feel like water potential is still a difficult topic and you would like to go over it one more time? I see one hand. Are there more people? Yeah, okay. Okay, so basically, water potential is a way to express the capacity of a certain solution or even air or soil, a place you could say, to attract water. So how can some place attract water? Maybe the pressure is really low and that may suck the water toward it. Or maybe the pressure is actually high and it pushes the water away from it. So pressure is one component of water potential. The other component, main component, is solutes. The more solutes you have in a certain place, the more water will tend to go there. And you can reason that by saying where there are solutes, there's no water. Water will go where the water concentration is lowest, so where you have the most solutes, the highest solute concentration. So a solute is anything that is dissolved. So those are the two main components, so pressure and solute concentration. And then there are some more minor components, which often are negligible. Those are if you have a lot of molecules that tend to bind water but are not really soluble, like if you have cotton, which is made of cellulose, then like cell walls may also hold on to water, so they may also attract water toward them, or soil may hold on to water. So that's called the matrix potential. And then a fourth component is gravity. If you get higher than 10 meters, that becomes a component as well. So the main two components, to reiterate, are pressure differences and concentration differences, which are measured as solute concentrations. They are the main components that contribute to water potential. And you can express those components as in a pressure unit. So the pressure unit that people typically use is megapascal, where 100,000 megapascal is 1 bar, which is atmospheric pressure. So 10 to the 5th pascal is atmospheric pressure. And we express it in megapascal, where megapascal is 10 to the 6th pascal. So atmospheric pressure is 0.1 megapascal. Yeah?
What were the other two negligible?
The negligible ones, matrix potential and gravity potential. So potential is shown as the Greek letter psi, psi, which looks like a trident. Water potential is shown as just psi or psi w. And the two components, solute potential is psi s. So that's the effect of the solutes. And then pressure potential is psi p for pressure. And the only equation you need to know is overall water potential, psi, equals psi s, solute potential, plus psi p, pressure potential. That's the only equation you need to know. And like I said, these components of water potential are expressed in pressure. They can be positive or negative. But solute potential is always either 0 for pure water or it's negative. The more solutes you have, the more negative it gets. So solute potential is 0 for pure water or it's negative. Pressure potential tends to be positive inside cells because there's a positive pressure, turgor, and it's negative in xylem because there's suction going on, transpiration from the shoot is sucking on the xylem. So that's really negative pressure, tension, a little bit of a vacuum. And then a flaccid cell, which is wilted, it has no turgor pressure, but it doesn't have any vacuum either, then pressure potential is 0. So pressure potential in a plant can be positive, negative, or 0. And solute potential is negative, because there are always some solutes in a plant. Pure water has 0 for solute potential. So the rule that's always true is water will go to where the overall water potential, so psi, is the most negative. So you can think of water always going downhill, always flowing downhill with gravity. That's way to remember. Water always goes to where the overall water potential is the most negative. So it can be most negative because of the pressure component, like in the xylem. But it can also be most negative because of a lot of solutes being there. So the solutes can be the overruling force to make water move across a membrane by means of osmosis, because the solutes kind of drag the water over that membrane into a cell typically. The solutes drive water movement into cells because they lower the overall water potential. And but if water moves out of cells, then it's typically the pressure that overrules the solutes and that push water out of cells. So we're going to, in this chapter, when we look at water movement in the whole plant, we're going to have a look at, we're going to follow water as it moves through the plant, and then we're going to look at each of these steps that water makes, into the roots, into the xylem, into the leaf, into the air, and then we're going to look at each of these processes, and we're going to say, so what's going on here? Do we have osmosis or not? If there's osmosis, is it solute-driven or is it pressure-driven that makes the water move across this membrane? So I was trying to give a recap of last module, and now we're going to start with the new module. Any questions about this recap? Okay, so we're going to have a look at water movement in the whole plant. And to reiterate, plants need water for healthy metabolism because all these biochemical reactions happen in water. And they need to have optimal solute concentrations for them to happen. And you cannot just have a lot of water lost and all the concentrations are off and things don't work anymore. Plants need also water to remain turgid, so support of the cells. And they need water for long distance transport. So in the xylem and the phloem, they have to have water movement to make that happen. So we're going to have a quick look at this long distance transport, although you have seen it when we did the recap about plant anatomy. So root to shoot transport happens by means of these red vessels, at least they're drawn here red, called xylem, and movement from photosynthesizing leaves to other plant parts goes by means of the phloem, which is shown here as these blue lines with arrows. And you see with the arrows the direction in which water moves in these vessels. So you see the red arrows go always away from the roots and they go to all the other plant parts. It's driven by transpiration. It's a negative pressure transpiration which sucks the water through the xylem. And the phloem movement you see goes away from mature leaves. So you see it starts in mature leaves and then it goes to all the organs that do not photosynthesize enough themselves, so they have to be fed. And that includes the young leaves. They do not photosynthesize enough yet, so they need to be fed. Also the fruits, they have to be fed with photosynthate, so sugars, and also the roots, they have to be fed, because all these organs do not photosynthesize at all or not enough. So that is the direction of the phloem transport. It goes from the mature leaves to all the other plant parts. And you see here that the fruit gets all its ingredients from the phloem, not from the xylem, because the fruit doesn't transpire enough itself to suck water up via the xylem. So the fruit has to be fed from the phloem. So it's just an interesting thing to realize. So phloem fluid is pushed through because of sugar loading in the mature leaves. This attracts water through osmosis. You realize now that this is going to happen. When you have a lot of high sugar concentration, this is going to attract water into that xylem in the mature leaves, and that's going to be pushed away through, did I say xylem? In the phloem. So the sugar loading in the phloem in the mature leaves is going to attract water from that leaf, and that's going to be pushed away through the phloem, away to other plant parts. So phloem is on a positive pressure, xylem on a negative pressure. And we're going to have a look in this chapter particularly at this red transport via the xylem. So we're going to have a look at how that works. How do people study these kinds of things? That's always interesting to look at. You can actually collect xylem fluid and phloem fluid. I'll show you some pictures of how that's done. But once you have collected droplets of this liquid, then you can analyze it. For instance, using some chromatography system, you can inject the sample into a system where you have a column that separates molecules based on charge or hydrophobicity or size. and then you get peaks, and then you can analyze what's in these peaks. So then you may get something as you see here. So the biggest difference you see here between xylem and phloem fluid is the sucrose content. You see there's half a molar of sucrose in phloem fluid, and there's no sugar in xylem fluid. So this clearly shows the function of the phloem in sucrose photosynthate transport. Other than that, you see that xylem and phloem both tend to have amino acids in there, and also some inorganic minerals like potassium, magnesium, calcium. So these inorganic nutrients are taken up via the roots, and then they can be mobilized via the xylem to different plant parts, and then they can, in some cases, be remobilized via the phloem. Not all minerals are equally mobile to be remobilized, and we'll get back to that when we look at all these different minerals that plants need. So minerals and amino acids we find in the xylem, and we also find them in the phloem, and we also find sugars in the phloem. And to give you an idea, 490 half a molar of sucrose, that's 180 gram per liter. That is a lot. That's with a cup full in a quart of water, so that's pretty sugary, the phloem fluid. And aphids actually feed by sucking on this phloem fluid. This is an aphid. And it sticks its mouth part, its stylets, right into a phloem vessel, the sieve element, as you see here, blown up. So this is the aphid mouth part, and it just can find this phloem vessel it sticks its mouth part in, and then through the positive pressure that's in the phloem, all it has to do is just let it flow out of there into its body, and then it uses those nutrients that are in there, and the excess it excretes here as little droplets from its butt, which it can actually, there's actually still a lot of sugar in there, and that you can sometimes find if you have your car parked under a tree with a lot of aphids in it, you can get all this sugary, sticky stuff on your car. And actually ants also milk aphids. They kind of tap them, and then all the sugary stuff comes out, and then the ants eat that. But anyway, that's a side story. So these aphids can be used in plants by plant physiologists. So what they can do is they can actually cut off this mouth part, and then this aphid has a hard time surviving probably. But what then happens is you see a droplet coming out of the mouth part that's still left sticking in the phloem sieve element, and that you can then collect with the syringe and you can analyze it. So that's how you can collect phloem fluid, by means of the aphid stylet method. And you can collect xylem sap in different ways. For instance, you can collect the stem, so you have to kill the plant for it, and then you stick it in this flask with a vacuum attached to the side arm, and that way you kind of suck dry the stem, and then you collect mostly xylem fluid, although it's probably going to be contaminated with some fluid, but it's mostly xylem fluid. So that's one way of collecting xylem sap. You can also push it out, like here. If you put the plant, and it can just be in its pot, just an intact plant, you put it in a sealed container and you put pressure on it, and then you push out the liquid, and it goes through the xylem, and it comes out at the end of the xylem vessel, from the tip of the leaves, at the end of the veins of the leaves, and then you can collect it. So then you get pure xylem fluid that's less contaminated with phloem sap. But you need kind of fancy equipment for that. Or you can do this, you can cut off the stem, and then especially if it's kind of humid and the plant has been well watered, then what you can see is that there's actually liquid coming out of what's left of the stem. And this is because there is root pressure from, especially in the morning, from roots taking up water and pushing it up. So there's not only suction from the shoot that sucks the xylem up, but there can also be root pressure pushing xylem fluid up. So it's a combination of the two. And especially at night, we get this pushing up of liquids, and especially during the day when the stomata are open, there's more suction going on. But you can make use of this root pressure effect if you cut off the stem, wait a little bit, maybe half an hour, and then you can collect the stem. If the air is not too dry, otherwise it would evaporate immediately, and then you can collect it. And this is probably going to be a little bit contaminated with phloem fluid too, but it's usable. So that's how you can collect xylem and phloem fluid. That's how you can analyze it. And just to reiterate, xylem is dead cells. It's just cell walls left standing. And phloem is living cells. You already saw that before. So now going to transpiration. So plants need water. Plants also, to some extent, need transpiration because they have to get the nutrients up to the shoots. So they have to have transpiration going on for that purpose. But another important reason why plants transpire, why they open their stomata, is because they need to take in CO2 from the air for photosynthesis. So that's the purposes of transpiration. So they have to bring water to the shoot, and particularly also nutrients to the shoot. And they have to, they open their stomata, which causes the transpiration to also take up CO2 from the air. When it's really hot in the summer, opening the stomata may also help the plant cool down a little bit, because transpiration cools down surfaces. As you know, when you're sweating, the purpose of that is so that you have evaporation of that sweat and that cools you down. So the same thing can happen to leaves of plants. That's a minor function, I would say. And to some extent, transpiration is also a necessary evil. Plants have to open their stomata to eat, you could say, to take up CO2 to make sugars. And so they cannot help but lose water in the process. But that transpiration also just has some functions, as you saw here. So how do you measure plant transpiration? There are several ways you could use. One is you put the whole plant on a scale, and you measure the weight loss, and that is a measure for transpiration. So you have to have the system sealed off, and then if you measure it over a reasonable time, so that weight gain due to photosynthesis is not really a factor, then you can measure weight loss over, let's say, a day or a couple of days. And so that is mostly due to water loss due to transpiration. So that kind of system is called a lysimeter, where you have a plant sitting on a scale, you measure weight change, and that's a measure for transpiration. And people have them at different scales. They may be as big as you can have even a little, like a vegetation, like a 1 square meter area on a big scale. And then that way you could try to mimic transpiration from like a piece of prairie land or something with a mixed vegetation. So you could have it at different scales, just one plant, or kind of a tiny microcosm or mesocosm or whatever people call it. So a little community, a little plant community. Yeah?
Does that type of thing work over like any significant length of time due to like growth and
Yeah, so the question.
water over and over?
Okay, so if you do this over a longer period of time, then it gets complicated, because then you may have plant weight gain due to photosynthesis. They take up CO2, make sugar, so they grow. And also you may have to water them. So then you would have to take all that into account as you do it. So using a lysimeter is easiest if you do it over a shorter time period. Those other factors are not a factor. You don't need to water them, and weight gain is negligible. So that's one way to do it. Another way to measure transpiration is using a little gas exchange chamber. There's a leaf chamber that you can put over a leaf, and you can then measure the water content of the air going into the leaf and the water content of the air coming out of the chamber. So it's a flow-through chamber. You put your leaf in the flow-through chamber, and you measure the water content in the incoming air and the water content in the outgoing air. And the difference between the two tells you what's going on here. So typically what you see is that there's more water in the outgoing air because the plant is releasing water. So from the difference between the two, you know how much water the plant is releasing per surface area of leaf, which you can measure. And then you can try to extrapolate that to the whole plant. If you know the plant's leaf surface area, then you can estimate how much this plant is transpiring. And then you can compare that. You can give plant different treatments, for instance, and see how gas exchange responds to that. And this water release from plants is mainly due to stomatal opening. So it's very much a function of, are the stomata open or not? Because when the stomata are closed, the plant the leaf doesn't lose much water at all. So I think at least 90% of water loss through leaves is by means of stomata. And that means that plants can also very efficiently save water by keeping their stomata closed. They will really only open them on purpose when they need to take up CO2 particularly. So that's the second way in which you can measure transpiration, a leaf chamber, a flow-through leaf chamber where you measure water content in the air that comes through. A third way in which you can measure transpiration is a potometer. And a potometer takes a branch or a stem, so you have to destructively sample the plant. You stick it in a tube, and the tube is filled with water. It's U-shaped, and you stick the branch or the stem in one end of the tube where it's in contact with the water, and on the other side of that U-shaped tube there's a little bit of air, and there's a pressure meter attached to that air. So when the plant, it's a closed system, this tube, so when this plant is going to transpire, it's going to suck on that water, and this gas that is going to expand because the water is going into the plant, so this gas is going to expand, and that reduces the pressure in the tube, and that's going to be measured. So the more the plant transpires, the lower the pressure gets, and the potometer gives you that pressure. So that's another way to measure transpiration by plants. So in that case, you use a whole branch, or you may even use a whole plant. And if you have a tree, another way you can measure transpiration is there are trunk collars that you can put around the trunk of the tree, and some of them stick into the xylem, and some of them just sense water movement by means of a thermocouple, I think. And so they measure the xylem flow in the trunk. So that's another way to do it. So the potometer is destructive, and the other three measurements are not destructive. Yeah?
In the lysimeter, like, is there soil in there too? And like,
Yeah.
how do you measure?
Is there soil in the lysimeter too?
Wouldn't some of the weight loss be just evapotranspiration from the soil itself?
Yeah, the question is with the lysimeter, wouldn't part of the water loss, so the weight loss, be due to just evapotranspiration from the soil rather than the plant? That's true. So that is, you could say, call it a complicating factor. That's hard to. Then what you would have to have, I guess, as a negative control is to have just an unvegetated same surface area of bare soil, and then subtract that from the vegetation. Yeah, good question. So that's how you measure transpiration, and that's why plants have transpiration. Now a little bit about what's going on during transpiration. What path does the water take from the soil water to the air if it goes through the plant? And which barriers are there? Which steps are taken by the water? And what is the driving force? So if we start right here, we have here a root with the root hair. And then we have here soil particles surrounded by soil water. And the first step that a water molecule will have to take is if it's going to pass through the plant and into the air, it has to get into the root. So it could travel actually in the apoplast of the cortex of the root, or it could go straight into the symplast, so pass a plasma membrane into the symplast and then just go from cell to cell via plasmodesmata, all through the cortex as well. So step one, if it is to go up to the shoot, it has to get at some point into the symplast of the root. And for that, it has to pass a membrane to go from the soil into the symplast, the inside of the cells of the roots. Then if the water wants to continue up to the shoot, it has to get into the xylem. The xylem is apoplast. But there is actually an impermeable layer that surrounds if you remember the vascular column that's in the center of the roots, there's this impermeable layer, the endodermis, Casparian strip, so water cannot pass through the cell wall there. That's why it has to eventually get into the symplast if it hasn't done so immediately. It has to eventually get into the symplast of the endodermis in order for it to then go through a second membrane to get into the xylem apoplast. So it has to go through the symplast at some point into the root, and then that's one membrane step, and then it has to pass a second membrane step to get into the apoplast of the xylem. 2 membrane steps. So that means osmosis. Osmosis is the movement of water through a membrane. Then once it's in the xylem, it will be sucked up with the transpiration stream. It goes with the flow. And there are no membranes in the xylem because it's dead tissue. And so our water molecules can go all the way up, bulk flow, and this is pulled up by transpiration. It's assisted by cohesion between the water molecules with the hydrogen bonding, and also adhesion of the water molecules to the sides of the xylem by hydrogen bonds as well. So this helps prevent the water column from falling down. So then all the way up in the xylem, if the water is going to continue, it has to leave the xylem, apoplast, and go through the leaf cells into the atmosphere. So then it has to pass another membrane step to get into the leaf cells. So that's osmosis. And then finally, it can pass out of the leaf cells into the intercellular space here within the leaf. And from there, it can diffuse out through the stomata into the atmosphere. So that would be another membrane step. So membrane step 1 was getting into the root symplast. Membrane step 2 was getting into the root xylem. Membrane step 3 was getting into a leaf symplast. And membrane step 4 is going through the membrane into the air. Initially, the air within the leaf, and then it can diffuse as a gas molecule into the atmosphere. And then in between here, we have this bulk flow step. No membranes here in the xylem. So that is how transpiration happens. And the driving force for all this, the main driving force, is transpiration here with the gas molecules going into the gas phase and leaving the leaf. That creates a very suction effect that pulls water up all the way down to the root. And in addition, there's a minor force for water movement in the xylem, which is a positive root pressure by water being following minerals basically in the root. Minerals through osmosis attract water, and this may create a positive pressure, pushing water up in the xylem. But that's more what happens at night. During the day, it's more transpiration. That is the driving force for water through the xylem. So to look more closely now, here we see the water movement or any movement of any molecule in the root. This is the cortex, this outer layer with the root hair and the epidermis. And then we have here the endodermis, which surrounds the vascular cylinder with the xylem and the phloem. And the xylem here is shown blue. So the xylem is apoplast. Cell walls are also apoplast. And the interior of the cells is symplast. And those different spaces here are, for convenience, color-coded. So the symplastic areas are blue, and the apoplastic areas are pink. So you see, if you are a molecule, then you can freely diffuse, travel via the apoplast. But if you want to continue into the xylem, which is also apoplast, you're forced to go into the symplast here at this level of the endodermis, because the cell wall of the endodermis is impermeable for water and solutes dissolved in water. So this is kind of a little raincoat. So any molecule that wants to go from the apoplast, that is the cell wall here, and the cortex, to the apoplast, that is the xylem here, in the central cylinder, is forced through the symplast here. It has to pass 2 membranes, one to get into the symplast and one to get back out. And those membranes give the plant control. So it can keep unwanted, maybe toxic elements out, and it can keep wanted nutrients in, so that there's no leakage. Question?
So an apoplast is basically just like an open halfway? Is that what it means?
Well, what apoplast means is that it is space in a plant that is not within a cell. And so that what that means is it's basically cell walls.
Within a living cell. Not within a living
Not within the plasma membrane of a living cell. That is what apoplast is. And so that's basically cell wall material. And cell wall material is not an effective molecular barrier. So molecules can freely travel through the cell walls, through all these fibers. The holes are big enough. So step one, at some point, our molecule, our water molecule, or any other molecule, will have to pass the membrane to get into the symplast, and then at the second step, it has to pass through another membrane to get into the xylem. And where that happens can vary. Where it gets into the symplast, it may get into the symplast right here at the tip of the root hair, or it may wait till the very end and go in right here at the level of the endodermis. And it can also happen anywhere in between. So step one, uptake into the root symplast. Step two, export into the apoplast. And if this happens for water, then it's membrane transport of water, and we call that osmosis. That is always passive, but it can either be driven by solute concentration difference or a pressure difference. In any case, the overall water potential, where the overall water potential is most negative, is where the water will go. That's always true. In the case of water movement into the symplast, then it's always the solutes that win out over the pressure, because the solutes are dragging water into the cell, because the solute concentration is always higher in the symplast than in the apoplast, but the pressure, on the other hand, is higher in cells than outside of cells. There's turgor pressure. So if water moves into cells, then those solutes win out over the pressure. So we call it solute-driven osmosis. But it's always true that the overall water potential is more negative where the water goes. So in this case, water potential is more negative in the symplast of the root than in the soil. That's why the water moves into the symplast. And the reason why the water potential is more negative is because of all the solutes. Because pressure is actually higher in the cell. So it's not because of pressure differences that it moves in, it's because of the solutes. On the other hand, the second step where the water moves out of the symplast of the endodermis and into the xylem apoplast, of course it's osmosis, it's water movement through a membrane. Of course the water potential is more negative in the xylem than in the endodermis, otherwise the water wouldn't move. But if you then ask, so why is the water potential more negative? Is it because of the solute concentration dragging the water into the xylem, or is it because of the pressure difference? Then the answer is it's the pressure difference that makes the big difference. It makes the water potential lower in the xylem, because the xylem has negative pressure. It's a straw that's being sucked on by transpiration. So that pressure difference is the reason why the water potential is more negative in the xylem, and that sucks the water into the xylem. And just a side note, the reason why we have all these solutes in these cells is because preceding water uptake, there has been mineral uptake. So all these minerals that have been accumulating over time allow for the water to then move in via osmosis. So mineral uptake is needed for water to effectively be dragged into that root by that plant. So once the water is in the xylem, there are no more cell walls, no more cell membranes, because it's dead tissues, it's dead cells. And then we call it bulk flow. If it's water movement, that's not really osmosis, there's no membrane. It's simply going with pressure difference. Pressure difference determines flow. That is bulk flow. And one case of bulk flow that I didn't mention is water movement toward the root in the soil. So this can also be caused by the plant effectively sucking up water, and this kind of drags more water toward the plants by bulk flow. And another case of bulk flow is here in the xylem, where the water is sucked up by transpiration in the shoot, and so this creates a pressure difference, and that makes that water move by bulk flow. It's not osmosis. And like I said, cohesion and adhesion of these water molecules with themselves and with the cell walls of the xylem helps in this process of water being pulled up and not falling back down. And it also helps that the xylem vessels are really narrow, so this makes the adhesion more effective. If the wider the vessel would be, the harder it would be to hold it up there. It would fall down more easily, but because it's so capillary, this helps. It's very thin. So bulk flow to the shoot, there's no membrane. We call it bulk flow, not osmosis, because there are no membranes. It's caused by negative pressure in the chute. Then we have step 4. We have again osmosis, because water is going to move into the symplast of the leaf. Water passes a membrane, and since it moves into the cell, it's the solutes that drive this process. It is osmosis, so the water potential in the cells must be more negative than in the xylem of the leaf. And it is the solutes in that cell that are the driving force for osmosis to happen, to make that water potential so negative in that leaf. And then some of the water molecules are going to stay in the plant because they're needed, but some eventually will probably evaporate, so that would be step 5 then. So then the water molecules go through the plasma membrane again, then they're temporarily sitting in the cell wall, which is surrounding these cells. And if you remember, especially at the bottom of leaves, you have this spongy mesophyll with lots of air holes in between them. So at this place, these water molecules may evaporate from the liquid phase, where they're embedded in the cell wall of these cells. They may go into the gas phase, and they fill this space. Typically, this intercellular space is 100% saturated with water. And then as soon as the stoma opens, here it's closed, but as soon as it opens, that water is going to diffuse out, because inside the leaf, 100% relative humidity. Outside of the leaf, probably it's going to be lower, depends where you are. If you're in the tropical rainforest, it may be 100%. Here in Colorado, it may be 5%. So the bigger that gradient in relative humidity, the faster that those water molecules are going to evaporate. This here you see a blow up of the cell walls, and you see kind of with this wavy line, water molecules going into the gas phase, so evaporating. But there's a little film of water kind of sitting here in the intercellular space. So as water moves out of the cells and into the cell wall, it passes a membrane, so it would be osmosis. And if it goes out of cells, then it's typically pressure-driven. So pressure-driven osmosis. So all along the way, as these water molecules move from the soil water into the air, the water potential goes down, 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 down all the way, as you see in this slide. So here we have a tree, a hypothetical tree. And this tree is 20 meters high. And we start here at the soil end. And we're going to follow the water molecule all the way out into the atmosphere. And here we have all the different places in the plants that the water passes along the way. And what we see in these columns are the overall water potential. So that is psi W here. So this, sometimes it's written as psi and sometimes as psi W. And then here you see the components that make up the overall water potential. And psi W equals psi P plus psi S, the only equation you need to know. And since this is a tree higher than 10 meters, we also have gravity in this case, which kicks in above 10 meters. So this starts here. But if you add all these three up, then you get this number. So a couple of things that I want to point out are, you see that the water potential gets more and more negative along the way. Otherwise, water wouldn't move. And another thing you see here is the pressure potential, psi P, varies, can be negative and can be positive. It's somewhat negative in the capillary spaces between the soil particles outside the plant, because there's kind of a suction going on toward the plant, because the plant is sucking up water. So there's a little bit of a negative pressure there. And then as soon as you're in the symplast, you see that the pressure potential is positive. That's turgor pressure. But then when the water molecule moves into the xylem, it's negative again, because in the xylem we have negative pressure, we have suction, because transpiration is sucking on the xylem, like on a straw. And you see that suction becomes stronger as we move from the root xylem to the leaf xylem, because that's where the suction action is. So it sucks the hardest at the top. And then we move into a leaf cell. So now we have a positive pressure again. We have turgor again. And then when we move out into the interior of the leaf in the cell wall, there's a negative pressure because evaporation is going on really, really strongly there. That's where the water molecules are escaping. So pressure can be positive or negative. In the xylem, it's negative. In cells, it's positive. And then here we see the osmotic potential, the other main component. And you see that this also varies, but it's always negative. It's never positive, because it's either zero if it's pure water, or it's increasingly negative with more solutes. And you see where it's the most negative is always in the symplast. So here we see the root cell and the leaf cell. They have quite a negative solute potential because they have the highest solute concentration within cells. And in the xylem, the solute concentration is much lower. It's much more dilute, and this is about 10-fold lower, as you see here. It's only minus 0.1. And in the soil water, it's also about minus 0.1. So that's just some solutes, some minerals dissolved in the soil water. Yeah, question?
Is there anything to determine whether, like, why the water would take an apoplastic or a symplastic route through the root?
The water will always take the path. Oh, so the question was, why would the water take the symplastic or the apoplastic route in a route? So I think the water always uses the path of least resistance. So whatever goes most easily.
It does vary. It's not like both routes are taken evenly.
I think it will fill up the space, and it will just go where there's negative pressure. What can I say? It will not leave a vacuum. It will fill it. But I don't know, like, oh, X% goes apoplastic and Y% symplastic.
Like why the pressure would vary, I guess. Like why if they're both consistently being used.
It will go wherever the water potential is most negative.
Okay.
So if you could say, well, the more solutes you have at that time in that route, the more the symplast is going to suck the water in, and the less it's going to stay around in the cell wall, probably. Yeah. Okay, so that was the osmotic potential. So hopefully you see that it makes sense. You see some pattern there. And then gravity kicks in above 10 meter, and you see it's a positive value, 0.1 megapascal per 10 meter. And the overall water potential is the sum of those components. And you already know that formula, psi equals psi p plus psi s. And in this case, psi g also is a component above 10 meter. So you can add them up in each case. And overall, as the water moves through the plant, the overall water potential becomes more negative, and that's the only thing that counts. That's the ultimate driving force. So you see solute potential varies, pressure potential varies, but the sum of the two shows a consistent tendency to go down and down and down. And that's what makes the water move. Another thing that is interesting to point out here is, look how low the water potential is in the outside air. That is really the driving force. The difference between the leaf internal air space and the outside air, that relative humidity difference between 100% relative humidity in the leaf and whatever, this is 50%. So that 50% is pretty reasonable. Already minus 95 megapascal, so that's the biggest difference in water potential along the way for this water molecule. So you can see how that can be the driving force for the whole process of water movement through the plant. So here now let's have a look at what the driving force is for the water movement in each step as it moves through this plant. So first we have soil movement toward the roots. This is bulk flow, so B for bulk flow. There's no membrane there, it's just we're in the soil. And it's the driving force is pressure difference. So the plant is moving water into it, and that kind of sucks more water toward the plant. The next step, the movement into the root cell, into the symplast, is solute-driven osmosis. It's osmosis because we have a membrane, and then there's always two possibilities. Is it solute-driven or is it pressure-driven? If it moves into a cell, it's solute-driven because all these solutes in the cell suck that water in. And then the second step, where the water moves out of the symplast of the root and into the xylem, then it's osmosis because there's a membrane involved. And then again, it's always solutes or it's pressure. In this case, it's pressure that makes that water move, because the pressure in the cell is positive, the pressure in the xylem is negative. Very big pressure difference. If it were up to the solutes, the water would stay in that route, because the solutes concentration is higher in the route. Still, the water moves out of those cells because of that big pressure difference. So pressure-driven osmosis. So whether it says S or P, it's both osmosis. It's just the question is it the solutes or the pressure that causes it. Once it's in the xylem, bulk flow, there are no membranes. So just the pressure, the negative pressure particularly that sucks that water up. Then we have movement into the leaf cell. That's, again, osmosis. We move across a membrane and into a cell that is solute-driven. Solutes suck that water in. And then if the water moves and goes into the intercellular space, then it's the pressure, the turgor pressure that pushes it out. Pressure-driven osmosis. So P and S, both osmosis, but into the cell, solute-driven, out of the cell, pressure-driven. And then you have just diffusion out of the leaf of the gas molecules, which you could call bulk flow. So hopefully you have an appreciation now of water movement through a plant, which membrane steps are passed, and that you have this osmosis and you have bulk flow. You have osmosis wherever there are membrane steps, otherwise it's bulk flow. And when you have osmosis, you have solutes that are a factor, you have pressure that is a factor. When water moves into cells, the solutes win, and that they drag the water into the cell by osmosis. And if you have water moving out of cells, the pressure wins. It pushes the water out of the cell. But the common factor is always that the overall water potential, where it's more negative, is where the water goes. So whether it's pressure-driven or solute-driven, the water always goes to where the overall water potential is most negative. And it can be most negative because there are really many solutes into cells, or it can be most negative because there is suction going on by transpiration. So pressure can be really low. Or on the other hand, you can have really high pressure turgor pushing it through, or both. Does it make sense? Anything still not clear that you want to ask or comment? No? Okay. Time flies. 2 minutes. So just to reiterate, driving forces for water movement in plants: short distance osmosis, into cells solute-driven, out of cells pressure-driven, long distance bulk flow, there are no membranes. Bulk flow can be due to suction. It's mainly due to suction created by leaf evaporation. And the movement up the plant is assisted by cohesion and adhesion of the water molecules to each other and to cell walls by means of hydrogen bonds. Root pressure is another minor factor, and we'll get back to root pressure later. Especially at night, root pressure is a factor. Okay. Be careful out there.